Welcome back to the Deep Dives, everybody. Today we're going to be exploring something pretty fascinating,
Yeah.
these compounds called secondary metabolites. And I think a lot of people hear secondary and think, oh, maybe not as important as the primary stuff. But as we'll see in this deep dive, these things are really, really cool.
Yeah, no, absolutely. And I think you hit the nail on the head when you said the word cool,
Ah,
because that's what makes this deep dive so
all
fascinating.
right.
We're going to kind of trace how scientists have figured out what these compounds are, what they do,
Mm-hmm.
and maybe even more excitingly, how we can use them now.
Right. Yeah. And we've got some great sources on this today. I'm excited to jump into it. So I guess to get started, the term secondary metabolite
Mm-hmm.
makes it sound like they maybe aren't that important, right? Like it's sort of an afterthought.
Hmm.
But can you kind of set the stage for us here? Like, what are we really talking about when we say secondary metabolites? And what makes them different than, say, primary metabolites?
Okay, so imagine a cell, right? Any living cell. And this cell is like a bustling factory.
Right. It's churning out all these different molecules. And these molecules are what keep the cell alive.
Yeah,
They're essential.
right.
So you've got things like sugars and proteins and fats, DNA, all the building
The
blocks.
essentials.
Yeah, the essentials, exactly. Now, those are your primary metabolites. They're the VIPs of the cell.
Gotcha. Okay.
But then you've got this other group of molecules being produced, and they're not directly involved in those essential processes like growth or reproduction.
Mm.
And these are the secondary metabolites. And
Hmm.
so for a long time, scientists thought, well, maybe they're just byproducts or waste products.
Yeah. Oh,
Like the cell's just kind of spitting these things out. But as we'll see,
Mhm.
it turns out they're far from being just junk.
Okay, so that's interesting. So at first we just thought, oh, these are just kind of there and they don't really do anything. But then obviously something changed that thinking, right?
Exactly. And one of the first people to really draw attention to these compounds was a guy named Albrecht Kossel back in 1910.
And he was actually a Nobel Prize winner. And Kossel, he was the first to kind of coin the term secondary metabolite and say, hey, these are distinct from those primary metabolites we talked about.
Yeah.
They're a separate category.
Interesting. Okay, so he was the first to kind of draw that line in the sand and say these are different. So what happened after that? How did our understanding kind of evolve from there?
Well, so for a while, people were still kind of scratching their heads about what these secondary metabolites actually did.
Mm-hmm.
But then in the 1930s, a scientist named Friedrich Sopeck came along, and he was really focused on plants.
Okay.
And he made a really interesting observation. He noticed that many of these secondary metabolites, especially those containing nitrogen, seem to be end products of metabolic processes.
Okay, and what does that mean exactly?
So it suggested that they weren't just waste products. The plant was actually investing energy and resources to make them, and that kind of hinted that maybe they had a purpose.
So there's a reason they're there. Okay, so from just kind of being there to maybe having a purpose, that's a
Yeah.
pretty big jump. When did we start to really figure out the specifics of what they were actually doing?
Well, like with a lot of science, the real breakthroughs came with advancements in technology.
Okay.
So, the mid 20th century was a huge turning point. We saw the rise of all these amazing analytical techniques like chromatography and spectroscopy.
Mhm.
And these are basically tools that allow scientists to separate, identify, and really study the structure of individual molecules.
So before this, it was just kind of like a soup of compounds and we couldn't really tell what was what?
Pretty much. But now with these tools, scientists could really start to isolate and characterize specific secondary metabolites.
Gotcha.
It was like they were suddenly able to see the individual trees in the forest.
That's a great analogy.
And then on top of that, we had this explosion in molecular biology and genomics.
Right.
And that allowed scientists to go even deeper and start understanding the actual pathways involved in making these compounds.
Hmm.
They could identify the specific genes responsible for their production, and they started to figure out the really intricate machinery that the cell uses to put these molecules together.
Wow. So we went from not even knowing what they were to being able to see how they were made at like a really fundamental level.
Exactly. And all this new knowledge really opened the floodgates in terms of practical applications.
Okay. So what were some of those applications? Like what did all this new understanding actually allow us to do?
Well, for starters, we realized just how many of these secondary metabolites there actually are. Like, nature is an incredible chemist.
Right.
It's churning out this massive diversity of compounds. And then we started to understand the really diverse roles they play in nature.
Yeah.
You think about things like the scent of a flower that attracts pollinators,
Right.
or the bitter taste of a plant that deters herbivores,
Hmm.
or even the antibiotics produced by certain microbes.
Right, right. And that's a huge one, right? Like penicillin.
Exactly. Penicillin is a classic example.
Yeah,
It's a secondary metabolite produced by a fungus, and its discovery revolutionized medicine.
absolutely.
It really highlighted the incredible power of these compounds to treat diseases.
Yeah. And it seems like that discovery kind of paved the way for a whole new way of thinking about how we could use these secondary metabolites.
Yeah, and it led to the development of some really cutting-edge technologies like
Mhm.
metabolic engineering and synthetic biology.
Okay, and what do those things let us do?
So basically, they allow us to manipulate and even design biological systems to produce these compounds on demand.
So we're not just relying on finding them in nature anymore. We can actually make them ourselves.
Yeah, exactly.
That's pretty amazing.
It's really about harnessing nature's chemical ingenuity.
Yeah.
We're not just observing anymore, we're actively participating.
Right. And I imagine that opens up so many possibilities, like in medicine, agriculture, all sorts of fields.
Absolutely. The potential is really huge.
So to kind of sum up, then we've gone from this basic categorization to understanding how these compounds are made to now being able to engineer them ourselves. That's an incredible journey.
It really is, and it's a testament to human curiosity.
Yeah, and I think the most exciting part is that we're really just scratching the surface,
Oh, absolutely.
given how many secondary metabolites are out there
Yeah.
and how little we still know about them. I mean, who knows what incredible discoveries are waiting to be made.
Exactly. There's a whole world of untapped potential out there.
And that's what makes this field so fascinating, right? It makes you wonder what amazing new medicines or materials or who knows what else might be hidden within these compounds just waiting to be uncovered.
It's like a treasure hunt, and we're just getting started.
And on that note, we'll wrap up this deep dive. Thanks for joining us, everyone.
Thanks for having me. It was a pleasure.
Until next time.